The lab tells us that having a shorter toe, big toe, than the secondary toe is dominant. So we can say that this would be our short big toe allele, and then this is a long big toe. So we have our three genotypes. Homozygous dominant is going to give you a short toe phenotype as well as the heterozygote. And then an only way to have a longer big toe than the secondary toe is to be homozygous recessive. So, I'll wait. So the Punnett square that they have you set up between two heterozygote parents is your basic monohybrid cross. This is going to give you a short toe, a short toe, a short toe, and a long toe. That's that 3-to-1 ratio, 75%, 25%. So they want you to actually do it. So you'll have 2 pennies or 1 penny, flip it twice. And if it's heads, right, if it flips heads, that's like getting the dominant. So the first parent will flip the coin, right? Since they're heterozygous, the heads would represent this allele and tails would represent that allele. All right, so if you end up getting the tails, it's the short. And then you'd flip it again. You'd flip the coin a second time or flip a second coin. It doesn't matter. Same scenario. All right, heads, you get a big T, would represent the big T. Tails would represent the little t. Now, so if you were to flip the heads one time, if you'd flip the coin, and you got 2 heads, right? That would be a big T, right? Yeah. And that's the same as having a short big toe. If you would flip it and it's got 2 tails, that's little t, right? And then if you got 1 heads, 1 tails, that's heterozygous short. So that's kind of the setup for the lab that you guys were supposed to do. Now I'm going to use sample data from one group. And they were supposed to sit there and just tally. How many times did you get 2 heads? You know, just count it and sit there and tally. You know, how many times did you get, heterozygous? Just sit there and tally. Just tally it up. And so you're going to get your total number of actual children, right? Oops, that's a little skewed. So I'm going to use a group's actual data. Like they actually went and had 100 children here. And so they had 27 of the individuals were 2 heads, so they were homozygous dominant, big T, big T. They had 55 times did they get heterozygotes, and then 18 times did they flip it and got 2 tails. Okay. And so the phenotypes, well, how many were short-toed? Well, if you add these 2 together, you get 82. 82 out of 100. 82 out of 100, that's 82%. On the long toe, well, they got 18 out of 100 children. That's 18%. Now, from the actual Punnett square, right, when you looked at the Punnett square, I'll draw the Punnett square for reference right here very quickly. If you remember, this is what you got. You should have gotten, right, 25% of your coin flips should have been that, right? Because that's 1 out of 4. 50% of your coin flips should have been this, correct? And 25% of your coin flips should have been that. So you predicted 25%. You actually got 27%. You predicted 50%. You actually got, this group did, 55%. Predicted 25%. They actually got 18%. Now the short toe, the actual phenotypes, continue on. The short toe was supposed to be 18%. I'm sorry, the short toe right here. Now we're talking about the Punnett square. I'm sorry, that's predicted. This is a short toe individual, and these are the long, I'm sorry, vice versa. My bad. The blue is the short toe because that's dominant, and the yellow is the long toe. So we're supposed to have 75%, or 3 out of 4. But this individual actually got what percentage? 82. The long toe, which was supposed to be 25%, that's the phenotypic ratio, they actually got 18%. So would you consider the predicted values to be the same, close, or not at all, if you looked at this data? I'd say it's probably pretty close. And the more times you flip the coin, the closer it's going to be to that 3-to-1 ratio. Now, part 2 had you flipping coins, but for this cross right here, big T, little t, little t, little t. And so, if you look at this Punnett Square, here's what you should see. Big T, little t, big T, little t, little t, little t, little t, little t. So this is short, and this is long. So for genotypic and phenotypic ratios, you're getting 50-50. Now, to do this with coins, you'd have to have one coin that has heads on one side and tails on the other. So if you flip it and you land heads, you get the big T, or the dominant for short. If you get tail, you get the little t, or for long toe. Now, for this individual, they're homozygous recessive. So what would happen is you'd have to have a coin that was either heads on both sides or tails on both sides. So does it matter whether you flip a coin for this individual here? What it's going to give? What is the only thing that this individual can contribute? A little t, right? So if you were to flip 2 coins to represent what allele you get to one parent, one coin you would flip heads, you would get the big T, tails, you would get the little t. The other coin you would flip, and no matter what side it was on, what would you get? Little t. So really, how many times do you have to flip a coin? Just once, to represent what this individual is going to give you, because this individual is going to give you a little t no matter what. So you flip a coin, 100 times. How many heads are you going to get? If you get heads, you get this individual. If you get tails, you get this individual right here, right? So I looked at the data of another group, and they flipped it 100 times. They got 52, 48. What percentage of your offspring were big T, little t? 52%. What percentage were short toes? Well, that is short, and that is long. It looks like tall, but it's long. 52%. This is 48%. This is 48%. Now, you're predicted from this Punnett Square right up here, you were supposed to get 50-50. You end up getting 52-48. And so your analysis, why do you think they are rarely the same as the actual? Because the predicted is just the possibility. Okay? The more times you flip a coin, the more times it's more likely to be what you actually predict. So then they ask you to do a Punnett Square, big T, big T, big T, little t. If you do that Punnett Square, no matter what are you going to get in terms of your toes? You're going to get 100% short toes. If you use the coin toss method to determine the ratios, would it come out the same or would it be different? You'd be different? You'd get less than 100%? It'd be the same. No, even if you flipped your coins, right? This guy's always going to give a big T, no matter how you flip the coin. Now, the other coin for this guy, half of them will be the big T and half of them will be the little t, but it doesn't matter. However you flip the coins, your actual results will be 100%, which would be the same as your predicted results. Okay? What do pennies or chips represent in the simulation? Your gametes, which contain the alleles. Each coin flip represents what the sperm will contain or what the egg will contain. Okay? When you toss the coin, and it lands up, you're actually simulating which process of sexual reproduction. This is the law of segregation. Which pretty much explains meiosis. And when you put the two flipped coins together, this is where you put where one coin, where a sperm meets an egg, then you get a fertilized egg, which is called a zygote. This process is fertilization. Right? That's what happens when you combine both flipped coins.